Let me just start by introducing Dr. Campbell, who is involved in the Vaccine Development Center at the University of Maryland. He's a pediatric infectious disease expert. Many of us know him from working with the Office of Vaccine Studies. I'm also really excited to mention that he has been named a co-director of what is essentially, I believe, the National Pediatric COVID-19 Vaccine Effort. And there's probably some official title to that, but that is probably a simple way of saying it. And so I think we are fortunate to have you with us and, to answer our questions and talk about the exciting, what I think is really, some light at the end of the tunnel here.
Great, thanks, Paul. And thanks everyone for joining in. I know it's a big group, and I'm glad to have my Frederick family on the phone to be able to talk to you. My family, like real family, asks a lot of these same questions of me all the time, and then folks at the CVD, people who are residents here, just everyone's media, etc. So, and I recently did a couple of town halls for the AAP, and a lot of the same issues come up. So I'm glad I have this long list of questions from the group, Paul, and I can give a quick overview of where things stand and then try to start answering some of those questions if you'd like. I'm sure I in some way related to Brenda Campbell, but I don't know exactly how far back it goes, somewhere back.
Hard to say.
Well, my thought was if you could just, like I had said, start with just sort of an overview of kind of the nuts and bolts of the vaccine and technology and why we, and your take on it and the key points. So.
Yeah, so right now in the United States, among the vaccines that have been in development and now two of which are you can divide them up into three types of people are now calling platforms. I think most of us who give, most of you probably who give vaccines, but you don't live in the world of vaccines, you don't think much about how those vaccines are made or what sort of type of vaccine other than maybe live and inactivated. But there's lots of different technologies for making vaccines. And if you split up the 6 kind of front runners, the 6 that'll probably be available to us, 2 which are now for adults and 4 kind of close, there's really 3 categories. The 2 that have already been authorized, so that's the one made by Pfizer, BioNTech, and by Moderna. Those are both the platform called mRNA. So M is messenger, and then you all know what RNA is. But messenger RNA is how essentially every life form transcribes what's in its DNA into the proteins that it needs to live. And it's the way that viruses make the pieces of their virus when you're infected with those viruses. But for the vaccines, what they've done, this technology, is take the mRNA, the genetic code, for just one protein within the virus called the spike protein. It's what makes a coronavirus called corona, because it has all these spikes on the outside, and so that gives it looks like it has a crown around it, and so that's why we call them coronaviruses. But so you just take that mRNA only for the spike protein, and it's put into these tiny little lipid spheres that are called nanoparticles. Because if you just give naked RNA, your body has enzymes that chop it up really fast. But if you give it inside the fat globule, then it'll be taken up inside of the cells before it can get chewed up. And then it gets expressed. So just like as if you were infected with COVID, your body will express that spike protein, meaning your cellular machinery will use the mRNA to make the spike. little bits of the spike, the whole spike, put some on the surface of the cell. Some will be what we call presented to the immune system by these special other parts of our immune response. Anyway, it acts similar to as if you were to be infected, but only that one little protein. You don't get all the symptoms. You don't get, you don't get really any of the symptoms of infection. You just get an immune response to the spike protein. That's the way both Pfizer and Moderna's vaccines are made. The two next ones coming down the pike are from Janssen, which is Johnson & Johnson, and AstraZeneca, or the Oxford vaccine. And both of those are what we call vectored vaccines. So what that means is instead of, like you all know about things like measles, mumps, rubella vaccine, which is the actual measles virus or mumps virus or rubella virus or chickenpox virus, and those are weakened. But what vectored vaccines are is you take a completely different virus, and in this case, adenoviruses, and you weaken them so much that they can't replicate. So it's like the shell of a virus, but it can't do any replication. And you put the gene for spike protein again into that other virus. So instead of putting it into a lipid nanoparticle, like for Moderna and Pfizer, you put it into adenovirus. One of them is a human adenovirus that we almost never see. We see a lot of adenoviruses, but these serotypes are the least common of all the adenoviruses. So you put the gene for spike into that, it expresses it on the surface, and then when you inject that into people, they don't get infected with the adenovirus and they don't get infected with coronavirus, but they make an immune response to the spike protein. So that's the next two coming. And then the two that are, the kind of fifth and sixth, I would say, in the running are made by a company called Novavax, and then by Sanofi Pasteur and GSK, which are, big vaccine manufacturers. Those are a little bit more traditional, meaning they are made by the technology which is recombinant DNA. So that's the same way, for example, like human papillomavirus, HPV vaccine, Gardasil, or hepatitis B vaccines are made. So you take the gene, again, that spike gene, you put it into something else, and in this instance, it's put into a virus called baculovirus, and then you grow that in the laboratory, and you use that virus in the lab as essentially like a factory. And then in these big vats, they make pure spike protein. But since injecting pure spike protein doesn't lead to a good enough immune response by itself, they add in an adjuvant. So both of those from Novavax and from Sanofi GSK are going to be recombinant spike plus an adjuvant, two different adjuvants. So there's something different about all of these vaccines compared to the vaccines that we currently have licensed on the market, and that is the two mRNAs. We don't have any mRNA vaccines on the market yet. The spike protein vectored ones, we also don't have vectored vaccines on, at least on the US market right now. And the two that are the recombinant technology is the same as Flublok and other recombinant vaccines, but the adjuvants are new. So there's going to be something new about all of the vaccines, but all of them also have either the platform has been tested over the last decade or so, or they have licensed vaccines or adjuvants outside the United States. So there is a history behind using all of these technologies. I mean, that's the big overview for where we stand. Two of them, as you know, the Pfizer and the Moderna, have gone fully through their phase 3 studies, which means their efficacy studies. And then those data were presented to the Food and Drug Administration. They reviewed it together with their independent body called VRBPAC. And then they recommended that it be authorized, and then it went to the CDC. That's the ACIP, is what we call their independent body. They made recommendations in who should get the vaccine. So the way it works for vaccines, as you probably know, is the first step is they get licensed or authorized. They get approval to be used and marketed in the United States, and then the CDC typically together with the AAP, the American Academy of Pediatrics, and the American Academy of Family Practice, et cetera, they make then the recommendations. Who should get it, what age, what dose, what kind of contraindications are there, all of that. So all of those steps have been gone through these first two vaccines, and in the next month to half a year, we'll have the same process probably going on with the other vaccines that are in the pipeline.
And Jim, I think a lot of people have had questions about, you know, how do we know that even though the process was, you know, so much quicker, that it's safe. So maybe you could talk about how, you know, a lot of all the same steps were done as usual, just a lot were done simultaneously, and how, you know, a phase 3 trial is a phase 3 trial, you know, regardless of, you know, when it's done in the process, and some of those questions, because I know a lot of them, you know, a lot of the questions were about the safety and how we can be sure of that.
Mm-hmm. Yeah, so there's a couple of reasons why the vaccine development process could go faster than it normally does. I mean, the number one reason is just because so many people at the same time with so much money, energy, manpower, et cetera, have been working together on this. Whereas, you know, most of the time there's some people developing one vaccine and another developing another one. And, you know, we've got a few things going on at the Pediatric Center that we're testing, and there's other places testing other ones, and they're all in this kind of, you know, slower, if you will, development process. Whereas when COVID hit, pretty much everybody that makes vaccines turned to trying making COVID vaccines, and everyone that does the clinical trials started doing clinical trials on it, and a huge amount of money was poured into it. And a lot of that money helped with the companies doing things that normally they wouldn't take the financial risk in doing if they didn't have that kind of money. So in other words, you know, would you as a manufacturer, before you even have the results of your phase 1 and your phase 2 trials, like the early vaccine trials, would you as the CEO of that company or anybody, you know, the board members say, okay, we're willing to put $2 billion into starting to manufacture the vaccine even before we have the clinical trials? No, you know, they wouldn't typically do that. But if the US government says, we'll give you that money, start manufacturing, you know, there may be a loss because if your vaccine doesn't work, then we're out that money. That's one of the main reasons why things have been able to move so quickly, is that a lot of people working on it, companies, developers, manufacturers, us in the academic world and all the trials places, and being able to start manufacturing the vaccines before we had the final results. I mean, another thing that made it go faster is the epidemic is raging, right? So when you have a disease that has very low incidence and you're trying to develop a vaccine and you want to see if it's efficacious, you have to wait a long time before enough people get infected after you've vaccinated them in a phase 3 trial before you'll know if it works. Because the way these trials work in phase 3 is some people get the vaccine. In this case, it was half the people got the vaccine and half the people get placebo. And then you just follow them over time and see what the incidence, like how many placebo recipients versus how many vaccine recipients get infected. And if the epidemic or the incidence is really low, it takes a long time to be able to see a difference, even if there is a true difference. But when you have a huge number of people getting infected, it doesn't take as long. So these trials for Moderna and for Pfizer, one was 44,000 people, half got the vaccine, half got placebo, and the other was 30,000 people, half and half. And when you compare that to typical vaccines that get licensure, these are on the large side. You know, we have some vaccines where they've been licensed in children with as few as 3,000 or 5,000 or 10,000. So it's, yes, they moved quickly, but no, it wasn't because they just said, oh, we've tested in a few people and we think it's safe and effective. It's, you know, tens of thousands. So you're on the same order of the number of people to be able to determine safety and efficacy. And the process, as you said, Paul, has been the same, just people met more frequently. You know, ACIP usually meets 3 times a year. I think ACIP has met 19 times, some huge number of times. Same with VRPAC. They say, okay, you're going to meet this Thursday and next Thursday. And so they've just met more frequently. They've reviewed things in an expedited fashion, but still with the same amount of data that you'd expect them to review. One of the reasons, people ask, why did our FDA take longer than, outside? Like the UK was a little faster and others were a little faster. The FDA actually goes to the raw data. They don't take the synthesized, analyzed data that's provided by the manufacturers and, quote, believe it. They take the raw data and they recalculate things like the efficacy and the proportion of people who had the different side effects. And they make sure that it jives with what's in what the manufacturer provided them. So that took a little bit longer, another extra week or two beyond what some of the other agencies did.
And also from a safety standpoint, my understanding is that with adverse events with vaccines, I mean, the vast majority of them occur during the first 6 weeks. I mean, at least that was what I've heard, and I've heard some others speak. So I think from a safety standpoint, the trial, some people say the trial, it wasn't long enough, but I guess it is long enough to know.
I mean, this comes up with every new vaccine we have, not just vaccines. It's actually still going on every time I give a vaccine talk, like a global vaccine talk, wherever. I get the question about, we probably still don't know about the long-term safety concerns for HPV vaccine, and people want to know, like, how can you assure me that 25 years from now, that if I give my son or daughter the vaccine, something bad's not going to happen? And, the question is like, how long is long enough? We know that these vaccines will be studied over the longer period, just like every vaccine has been. We've never had a vaccine where a decade later we found something that we weren't, that was something odd that then a decade later is going to be some problem. You can never say that that couldn't happen with a vaccine, but we've been vaccinating people for a long time, and just so the reason why people say like 6 weeks is mainly is due to the definition people used for vaccine-associated Guillain-Barré syndrome, right? So where back with swine flu back in the 70s, and there was some GBS associated with it, the definition was if it occurred in the first 42 days after vaccination, we thought it might be due to the, it could have been triggered by the vaccine. I think that's reasonable, somewhere on the 6 week, 2 month, 3 month, and you all vaccinate people all the time. I'm sure you're not thinking, oh, something that happens to a child 6 months after they got their vaccine might be vaccine related. mRNA is gone in about 3 days. So whatever happened, all the making of that spike protein that happens in the cells is happening in those first couple of days after you get the vaccine. And then there's no trace of that ribonucleic acid anymore. So the rest of what's going on is your immune system, responding to that, and your T helper cells telling your B cells to make antibodies, et cetera. So there's no vaccine product left. I don't think we have any reason to believe that there's some other thing that might happen with these vaccines. The very first person to get the Pfizer vaccine was here at the University of Maryland in May. So we have about 7 months worth of follow-up for that person now. And the FDA wouldn't accept an application from the manufacturers for their phase 3 data until they had 2 months of follow-up for the people in the studies. So those people are still in the studies and they're continuing to be followed. It is, things have been expedited, but I don't have special concerns about some long-term safety about these vaccines versus others.
Yeah. A lot of questions came up about allergic reactions. So I didn't know if maybe you could address that, especially because that's what gets into the, sort of into the news.
Yeah. I mean, usually what goes on with any vaccine is there are a small number of people who have allergic reactions, some of which are mild and some of which are more severe, and they happen behind the scenes and there's no media associated with it. But because it's COVID, everyone knows about what's going on with these. I think we still don't know for sure whether or not the rate of severe allergic reactions will be the same, less, or more. But what we do know is that in the trials, the rates were no higher than expected, and essentially no severe early anaphylaxis. I was, when I told that to my wife, she's like, oh, but the New York Times here says that there were 2 cases of anaphylaxis in the Moderna trial. And that's true, but they weren't right after people being vaccinated. So it's a little bit unfair for the media to say it that way. One of them was 63 days after the 2nd dose. So, how do you posit that the vaccine 2 months later led to an immediate type hypersensitivity reaction? It wasn't. And the other one was between the 2 doses, somebody got contrast, for an X-ray study, and they had anaphylaxis associated. So they were in the trial and they had anaphylaxis, but the chances of it being due to the vaccine were very low. So does that mean, usually what we say for anaphylaxis and vaccines is that it's less frequent in general for vaccines than 1 in a million. I usually say 1 in 4 million when we do our studies generally. It probably depends on the vaccine. I don't know yet whether for these vaccines it's going to be still in that rate. We haven't vaccinated that many people yet, but there have been some who have had severe reactions. And right now, the recommendations from the CDC are that it's contraindicated for people who have a known severe allergic reaction to something that's in the vaccine. But it's unlikely you're going to know that before getting one of these 2 vaccines, because there's not much in the vaccine that you could know that you were allergic to. In other words, I don't think anybody knows they're allergic to an mRNA product for spike or to a lipid nanoparticle. The only other things in there are sugars and salts. So I think until you get the first vaccine, there's no way to really know if you have an allergic reaction to this vaccine. And then there's precaution for people who have had a severe allergic reaction to another injectable medicine or vaccine. And for those, it's recommended that you kind of weigh how bad is, either how bad were those reactions in the past and how much at risk do you think you are for getting COVID-19. I think if you're at really high risk, you're going to say it's worth it. If you're not at high risk, then maybe you would want to wait until you know for sure that there's not something about the vaccine that we don't already know. There's no latex, I can tell you, in either of the rubber stoppers. There are no adjuvants in these 2 vaccines. There are no preservatives, and there are no antibiotics. So those sorts of things that are the typical things people might be allergic to, none of them are found in the vaccine.
A couple of questions came in sort of related to sort of the immunity of the vaccine. How long it might last, will it be annual, which I'm sure some of these, we're still finding out, but also there's also this idea that the vaccine, at least the mRNA vaccine, may even provide better protection than natural infection. So maybe you could comment on what we know about the immunity of the vaccines.
Sure. So we think, what we in the vaccine world, it's important for us to think about what is the most important part of the immune system that we ping, that we hit for different infections, because different infections are protected by different arms of the immune system, or by different what we call correlates of protection. And it looks like for COVID-19 that it is what we call neutralizing antibodies against the spike protein. That's the most important part of the immune response. And by neutralizing, we mean not only does it stick to the virus, but that when sticking to the virus, it prevents it from going cell to cell. And for both the animal models, for previous coronaviruses, and now for this one, it looks like that's the most important thing. So in the trials, what people have shown is that the amount of neutralizing antibody that volunteers in the study have reached, the height of that antibody, has surpassed the amount in control, what we might call controls, who have recovered from SARS-CoV-2 COVID-19 infection, what we call convalescent plasma. So we think that being infected leads to some amount of protection. We don't know for how long and how good that protection is, but it's probably pretty good early on, and it may last a long time. We don't know that yet. But the fact that by being vaccinated, you get higher antibody responses means you may get actually better protection from the vaccine than you do from the natural infection. I can't say that for sure yet, but it's possible given how high the antibody responses were. And we don't have a number for people yet to say, okay, if you got infected, you're, for example, 95%, that's 95% effective against repeat infection. But we do know from the trials exactly what that efficacy was, 94.5 and 95%. So, you know, it's like the sure thing, the vaccine is surely protective because of those trials. The infection, I think, is very likely also to be protective, at least in the short term, but we can't put a number on it. In the trials, not everyone, but a proportion of the people in the trials had previously been infected with COVID. Like in one of the trials, like 2,000 out of the 30,000. So we know it's safe to get the vaccine if you've previously been infected. And what probably happens is your immune system the first vaccine that you get actually boosts, and the second one you get boosts even more, rather than what happens if you've never been infected before. Then it takes that first one to what we call prime the immune system, and then the second one to give the boost. So just like with other infections, getting a vaccine after having had the infection, for example, getting a chickenpox vaccine after having chickenpox, or getting, measles vaccine after having measles, anything, there's no additional risk to getting the vaccine, but there is the potential of kind of boosting the immune system and giving you even better protection than you get from the natural one.
Yeah, that's great, because that was certainly a question that came up, and I've also heard that there's no reason to get tested before you get the virus, before you get the vaccine, or if you've had it before, to worry about.
I mean, there have been some things out there with people saying, like, if you are authorized, your number comes up to get vaccinated and you recently had COVID, and that people may choose to say that they will allow someone else within their phase to get vaccine, but that's a different question really than whether or not it's safe and would be effective. Yeah. And like you said, Paul, it's not recommended that you get tested if you're not sure if you ever had COVID. And if you know that you've had COVID and you have the opportunity to get vaccinated, it's certainly fine to go.
It's just going to boost your immunity, really. Right. What about if someone doesn't get the second dose, or do we know about the second dose, in that a lot of vaccines, if you're late for the second dose, even if you're really late, we don't repeat the first dose. Do we suspect that's the same with this vaccine?
Yeah, I mean, we don't know yet, because as you can imagine, in most of these trials, people are fairly motivated, and so are the investigators. And you guys know from our trial there, we do our best to try to get people on time within the window and get in. So the majority of people hit pretty much on target, get the vaccine as expected. So it's not known, but it's not. It's the same. The recommendations are the same, and that is if you miss that time, which is either 21 or for these two vaccines, 21 or 28 days for the second shot, that you just get the second shot whenever that is, and it will likely boost it. If you just take historical precedents, it's actually likely that if you're a little bit late, you'll have a better response, because some time between those vaccines typically gives you a better boost. That's one of the reasons that, for example, we wait a fair amount of time to boost kids. Like, we give, for example, the primary series 2, 4, and 6 months. And then we say the next one, if you're giving a booster, you got to wait until after the first birthday. It's because giving the immune system that extra time gives you a better anamnestic booster response. I mean, we don't know that for COVID, but if it's the same as other vaccines, then it's not going to be so bad if people are late. The main problem with being late is you may have waning between the first and the second. So you may be at higher risk, you know what I mean? So you get the first one, and let's say you don't get the second one until 6 months later. Well, maybe in month 4 and 5, before you got that second one, you've had like waning immunity, and now you're not protected in that period. So really, all we know for sure is that 2 doses leads to this 95% protection. There is some evidence, although it's not as strong yet, that even after the first dose, at about day 10 or so, people are starting to see protection. Because if you look at the data, what you do is you look at the curves, meaning the incidence, like how many people got infected over time in the 2 groups, the one that got placebo and the one that got the vaccine. And it's a really impressive graph where the people who got the vaccine in the first like 7 days, they go up at the same rate as those who got placebo. As soon as you get to about day 7 or 10, the people who got the vaccine, it just flatlines. It's just almost the same all the way out to 3 months, meaning very few people after day 10 got infected. But the placebo group, it just linearly just keeps going up, 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 up. And the lines get further and further apart the farther from vaccination you get. So in those first week or so, no difference. Those people were probably already incubating at the time they got vaccinated. But as soon as you hit about 1 week or 10 days, the 2 curves split and they get further and further apart the longer you wait.
Now, I mean, we have a lot of questions and there's a lot of interest about, what's the plan for the pediatric age group and pediatric vaccine development. But before we get to that, just a couple other things about, the vaccines and safety. One question was about, antibody dependent enhancement. And the other was also about, this burning question about if you get vaccinated, can you transmit the virus to someone else?
So I think for these vaccines, the 2 of the major, theoretical safety concerns that we don't have any evidence for, but that we're all, thinking about and want to make sure that we never do have evidence for them. One is what you brought up, Paul, which is what people now call vaccine-associated enhanced respiratory disease, meaning could it be that you get the vaccine and instead of protecting you, that when you're exposed to COVID, you actually get worse disease than if you never had gotten vaccinated. That's really only happened with one vaccine before, and that's the killed RSV vaccine back in the 1960s. There are some other examples of things that are similar, like the killed measles vaccine in the 60s and dengue vaccine, but it's a little bit of a different story with the dengue vaccine because of different serotypes. But we have some ideas about what the main causes of that problem or those problems are. And one thing is which way the immune system is pushed, if you will, when you're vaccinated. And we separate out these sort of like 2 ways that your T cells go. One is called Th1 and the other is Th2. And Th2 are allergic type reactions. So those are like when you get anaphylaxis, when you get any kind of allergy, eosinophils, IgE type direction. IgG and neutralizing and those sorts of protective effects are typically Th1. So as these vaccines were being developed, everyone was looking closely at which way, is it a balance or is it going? These are all Th1 skewed vaccines so far. So that's good. There's no biological, like laboratory evidence that we're skewed the wrong way for enhanced disease. Then in animals, these have been tested in animals. So what you can do in an animal is you can purposefully infect them and see, do they get enhanced disease? It was not seen in that, no enhanced disease in animals, even including non-human primates, meaning like macaques and monkeys that have, think you can get like COVID-like disease. The second thing is whether or not you make a lot of what we call non-neutralizing antibodies. So these are antibodies that your immune system generates, but they don't neutralize, they just stick. So they're binding, but non-neutralizing. And again, the like preclinical evidence, and now the clinical evidence is pointing skewed towards high levels of neutralizing antibodies. So both of those make us think it's not likely that we would have enhanced disease. And what you've seen so far in the adults is just the opposite, right? Not only is it protecting the adults against mild and moderate disease, but it has very good protection against severe disease.
Right.
So in one study, there was only one severe case in vaccine recipient. In the other study, there were none. All of the severe cases were in the placebo recipients. So it's not pushing the adults to get more severe disease, it's just the opposite. So could it be different in children? It's always possible, but it's not very likely.
Right.
The other thing is MIS-C, like people are bringing up, could the vaccine either trigger MIS-C itself, or could it mean that when you're exposed to SARS-CoV-2 in the future, if you've been previously vaccinated, would you have a higher rate of MIS-C? Again, there's just no way for us to know that before we do the studies, but I think it's very unlikely. I think the most likely thing is the same thing that happens with other vaccines, and that is if you can protect someone from getting the infection, the disease, you're likely to protect them from getting the sequelae of the disease. So if MIS-C is a sequela, a follow-on, an abnormal immune response after being infected, and you can prevent people from getting infected, then you prevent them from having those abnormal post-infectious problems. Again, we don't have a good biological marker for MIS-C, so until we do the studies, I think we won't know for sure. There is MIS-A, which is like in young adults, like 18, 19, young 20s. Some of those people have had the same type of problem as MIS-C, and there were no cases of MIS-A in the trials. So I think that's encouraging.
Right. And in terms of, if you got the vaccine, can you give it to still be infected, give it to someone else? Maybe you can comment on that, although.
We don't, again, that wasn't the primary outcome measure for the trials. So what happens in these trials, as you all know probably from the trials that we do in Frederick, is you have a primary outcome measure, and that one is the most important question you're asking. And the most important question to be asked for these was prevention of laboratory confirmed symptomatic COVID-19. Because public health wise, individual protection wise, most important question to ask. But one of the other questions being asked is, will it prevent asymptomatic infection? So just getting the disease, getting the infection without having any symptoms. We don't know that yet. Again, from animal models, it looks like it does prevent infection. There is a little bit of evidence in these trials that it may prevent infection, and that is in the Moderna trial, which was the evidence, the data were just presented this last weekend, well, end of last week and then into the weekend for FDA and CDC. They presented in a supplement to their application an interesting little piece of data, which is they did a SARS-CoV-2 PCR when people were enrolling, so day 0 before they got their first vaccine. And then they routinely, without any symptoms, just the day they came in for their second vaccine, which was 28 days after the first one, they swabbed everybody's nose then also. And there was a 2/3 reduction in asymptomatic infection on day 28 after having received a single vaccine. So 3 per 1,000 people who got the placebo were positive on day 28, no symptoms. 1 per 1,000 were positive who had gotten the vaccine. So 67% reduction. Is that proof? Not really strong proof, but it certainly goes in the direction of making us think that it's likely that it does prevent infection. Is it going to prevent all infection? Probably not. Most vaccines prevent a certain amount of infection, and then maybe more mild and moderate disease, and then more severe or very severe disease. So if we had to guess, you know, we're going to get 95 very high protection rates against moderate to severe illness, a little bit less to the like posse symptomatic, as we call it, and then maybe a little bit less to overall to infection. And why is that important? First of all, when people get vaccinated, they still need to do all the same things that we're doing now. That is, you know, face masks and social distancing and hand washing and et cetera. And until we know the answer to that question, we're not really going to know about how much herd immunity the vaccine will lead to. And herd immunity, for those of you who don't live in this world all the time, it means, you know, when we vaccinate populations of people, or when populations get infected, you start getting fewer and fewer people in the population susceptible. And when you get to a certain number who are non-susceptible, then even the susceptibles have an amount of protection just because the chances of them being exposed to the virus get very low. And the more contagious the pathogen is, then the higher the number that we need in the community to be vaccinated or non-susceptible. For this, it's kind of moderate amount of contagiousness, if you will. It's much lower than like measles and things like that. It's higher than some others. Probably around 70%, maybe 75% of people in the community need to be vaccinated or non-susceptible before we're going to see any amount of reduction in the disease due to, quote, herd immunity. Now, individuals may be protected, but the community protection won't occur until we get the pretty high rates of vaccination.
And then again, I want to leave a couple minutes for the pediatric focus, but for our staff and people here, we now, you know, I've been here a long time, so it's not true for me anymore, but it does seem like there are always a lot of people at the pediatric center that are starting a family. Yep. And a lot of questions did come up about, you know, safety in pregnancy. I think people worry about, you know, what we know about breastfeeding. There was, I think, some report out of the UK or some social media thing that went viral about infertility. And I was, you know, and a lot of our staff are certainly in that stage of life where they're starting their families or have young families. So.
Yeah, so let's go from maybe the top to the bottom there. So first about the infertility issue. If you're concerned about that, I really suggest that you don't just read look at what some of the things on social media are, but look more deeply into it. But I'll explain it to you a little bit about why it's even out there. And start off by saying, no, we do not believe that this vaccine could possibly lead to infertility. So what's the theory behind it? Proteins in the natural world, whether they come from bacteria or plants or animals or whatever, there's sometimes what we call homology. So those proteins, as you might expect, may have some similarity to human proteins. And so if you make an antibody against a foreign protein, and that foreign protein is very similar to a human protein, then maybe that antibody is going to attack your own, your own organs or cells. So not unexpectedly, if you blast, meaning like you go out there and look at what the gene structure of spike protein is, you can find on the placenta. It's very small homology. There's lots of homology with lots of different proteins. It's very unlikely that any antibody response you're going to make to spike is going to have any effect on a placenta. So there is no evidence, other than somebody doing that blast search, that there's going to be any problems with the placenta. Now, what other evidence do we have? Well, when you get infected with coronaviruses, both the endemic, we have four coronaviruses that just cause the common cold. You make anti-spike antibodies. Coronaviruses have not been associated with miscarriage or infertility in any way. Neither has COVID-19. There are studies now looking at this virus and does it lead to that problem among women, early pregnancy loss? No. So if the natural infections don't do it, and if the only thing we're talking about is this theory about this tiny little homology, you can find homology in so many different things. It really is over. I think it really is overblown. So I would not get too deep down into that rabbit hole.
And the pregnancy questions?
Pregnancy, we have very little data on pregnancy so far because pregnant women were not allowed in the trials when the trials started, but there were pregnant women, some dozens of pregnant women now, women who became pregnant after starting the trials. No concerns for adverse events among them, but that's very little data to go on. Yeah. ACOG, which is the American College of Obstetrics and Gynecology, they just came out with a nice statement on what's recommended for pregnant women or women wanting to become pregnant and women breastfeeding. And I think reading that would be really helpful for people to understand where people are coming from. The overall gist is if you're breastfeeding, ACOG and everyone else thinks you should just go ahead and get vaccinated if you're in the list to get vaccinated. If you're pregnant or wanting to become pregnant, then you kind of have to decide based on how much data we have, which is very little, the theoretical risks, which I think are very low theoretical risks, and your risk of becoming infected, and putting those together with your family and your provider to make the decision whether you get it. The main gist of the whole, both what ACIP has said and what ACOG is saying is, if you're a woman who is pregnant or who wants to become pregnant soon and you are eligible to get this vaccine, we as recommending bodies should not be telling you that you cannot, because you need to make the decision for yourself, like, am I at high enough risk? I mean, we have women here who are pregnant, they work in the ICU and they're caring for COVID patients all the time. And many of them feel very strongly like, don't tell me because I'm pregnant that I can't get a 95% effective vaccine when my risk of myself getting infected and maybe getting very severely ill is high. So I think, I know that's hedging a little bit, but without additional data, we can't say more about different trimesters and those sorts of things.
And I'll try to track down that ACOG statement and get it out to everybody.
I know Nancy has it. We provided it to someone who asked within the space. She did.
And then in terms of the, what's up for our patients in the future and what's happening, I know that there's some questions about, it's been used down to 16 and now it's being studied in 12 and up, and what does it look like in the future for us?
Yeah, so essentially now all of the manufacturers that are further along either have already or are moving down into teenagers. So Pfizer already has studied it down to 12 and they're continuing to do that. When they put in their emergency use authorization, asked for authorization down to age 16 and they got it. So they're authorized to age 16. And when they put in for licensure, which will probably be in April or May, they're going to put in down to age 12 is what we've heard. Moderna has started their study down to age 12, but none of the results yet. And Janssen is about to start down to age 12. What we will be involved in, our group will be the young children. So probably we will be doing the Janssen study, and that first group is 3 to 11 years old, then 1 to 2 years old, then and what we call age de-escalation.
Okay, one second. Somebody must have turned their microphone on. So if you can mute, thank you.
So when will those things begin? We're hoping February. I've been working since the summer on writing the protocols, trying to get the manufacturers to agree to move forward, trying to get the NIH to move forward. I've testified before like the National Vaccine Advisory Committee and before the Red Book Committee and before lots of different people to say like we need to move this along. And we've gained traction over time, but it has taken some time. I was in the camp of let's move this a little more quickly. Some people wanted to move more slowly, but everything is on the move now. So our expectation is early in 2021, we will start these trials. And what I'm pushing is to try to get results, at least in the older children, like school age and teenagers, that we would be ready for next school year, that we would have at least a vaccine, but maybe 2 or 3 vaccines, just like we have for adults. Ready for kids that we could vaccinate in the late summer before they go back to school. Now, who knows? I mean, I can't tell you for sure that's what's going to happen, but that has been my goal. Is at least to get that. We thought we were going to have already started, but it's taken a little bit longer. People have been focusing on the adult trials.
I mean, I do think for at least for our patients. The good news to tell parents is that, especially in the younger kids, I mean, there certainly can be some bad outcomes with COVID, but especially in the younger kids, and even up to 16 or 18. I mean, the amount of disease related to COVID compared to, the older population is really, it's such a small group, but it's not nothing, and I think we need to protect our patients, but at least we could reassure them. From that standpoint?
Yeah, I think that is true, especially when comparing to, either adults with comorbidities or older adults, that obviously the morbidity and mortality is a lot lower. I mean, our push has been to say, but if you compare this to other vaccine preventable diseases in children, that it ranks right up there, right? Because
I think the number of deaths from COVID is about the same as there were in flu last year. Is that correct?
Yeah. So it's been a bad flu year, but it has been an entire year's worth. So, and thousands of kids who've been hospitalized from it. And my other, reason why I think it's very important is that even if kids are not bearing the brunt of the burden of the infectious complications, they are bearing a huge societal burden, right, with this pandemic. You all know that, we know that, in terms of, no school and no grandparents, no play and all of that. If vaccines would allow them not only to get protection against the infection, but protection against this societal burden, it's going to be, important.
Yeah. And do you have any advice for our patients who we have, it's a big practice. We have a lot of patients who are, even 16 and older with underlying health issues, immunocompromised for whatever reasons, congenital heart disease. Any advice we could give to patients about where they're going to be in the strata of vaccine recipients?
Yeah, they will, the thing is it has to be authorized for their age, but once it's authorized, they will be within the same, phased in the same way as for adults. So for the Pfizer vaccine right now, where it's authorized down to age 16, right now, because we're still in phase 1A, the only 16 and 17-year-olds that would be eligible, well, there probably would be very few, but the thought was the only ones who might be eligible are those who are going to be working in the healthcare setting, even at age 16 or 17, because healthcare providers, not just providers, it's healthcare people who work in the healthcare setting. But then the next level down, so the 1B, the one that's coming very soon, will be frontline essential workers are added into that. And some 16 and 17-year-olds might fall into that category. But the majority that will be eligible won't be until we get to those with the that we get to comorbidities. So it's not far off, but it's probably I mean, it's not immediate, but it's not that far off. So probably months from now. And I think both Moderna and Pfizer are going to be able to put in for their full licensure, probably are going to put into down to age 12.
Mm-hmm.
Then we'll have all those teenagers with comorbidities will be eligible as long as ACIP says that they can get it.
I know we're running out of time. Just to get to a couple of questions that we just didn't get. Actually, one interesting question was for people who were in the vaccine studies that got the placebo, are there plans for them to get the vaccine?
Yeah,
that was one.
That's a hot topic. Lots of controversy surrounding that. Both VRPAC meetings, they lasted from like 9 a.m. to like 6 p.m. And I would say 2-3 hours each time was spent on this exact topic. They had an ethicist from Stanford who talked about the topic. They had the company saying what they want to do, the FDA, VRPAC, everybody. So yes, there are plans. They're slightly different, it looks like, for the Moderna and the Pfizer studies, but they are both planning on either unblinding or allowing people to get vaccine. There's a couple of different ways that it can be done. What the ethicist from Stanford was recommending, I think it was an interesting twist, is don't unblind, but anybody who becomes eligible in the community, because they're in a phase where they could get it outside of the study now. You say, we will offer you to get 2 shots, but we're not going to tell you whether you first got vaccine and now we're giving you placebo, or you first got placebo and now we're giving you vaccine, but we'll assure you that once you get those 2 shots, you've been vaccinated. And then you keep allowing them to, in a blinded fashion, to keep receiving vaccine as they're allowable in the community. The companies argued back that that would be logistically very difficult and that it would delay, and there are a lot of healthcare workers in these studies, and they'll just leave the study and we'll lose all of them. So I think what's mainly going to happen is they're going to allow unblinding and keep people who want to stay blinded for as long as they can. The main reason to keep the blinding is to see more with side effects over time, whether or not there is any difference in these months. Other than that, there's not a lot of reason for them to be blinded for much longer. Originally, they are still going to be in the studies for 2 years, and the thought was they could be blinded for 2 years. That's not going to definitely not happen. And the pediatric study, our plan is most of the kids will get the vaccine, an 8-to-1 ratio, but the one that get the placebo, they will quickly be converted to vaccine. They won't know, they'll be blinded, but within the structure of the study, they will then get vaccine. So every family will know that the kids will get vaccine. It's just a matter of, are they getting it at the beginning or are they getting it a little bit later in the trial?
Right. I mean, I think the one question that just I know wasn't answered that I see on our list, recommendations for people who are immunocompromised or have some type of autoimmune disorder. Are there any recommendations that they should not get vaccinated, or?
They're
I would
not.
think we want to protect them.
Yeah, the only contraindication is severe allergic reaction to a component of the vaccine. There were not a lot of people with immune compromising conditions, or at least severe immune compromising conditions in the studies. There's no reason to believe that the safety would be any different. It is possible that immune responses might not be as good among the people who have immune compromise, depending on what the compromising condition is. But you know, those same people are at higher risk of really any infectious disease. So I think I would seriously consider, if you can get 95% protection as a non-immune compromised person, you probably can get close to that as an immune compromised person. That it's probably worth considering getting.
Is there any information on different responses in different age groups? Like I did hear that maybe some side effects were a little bit greater in the teenagers that got it, thinking that maybe kids need a lower dose, or did older people respond just as well as younger people?
In terms of efficacy and immune responses, they were about the same across the ages. So even in the very old, people were worried that in the very old, that they would have poor immune responses, but it turns out that wasn't the case. Now, there's not huge numbers in these trials, but like people over 75 had 100% efficacy, but there were only, I can't remember the exact number, something like no cases in those who got vaccinated and 7 or 8 cases in those who got placebo. But it still looked like it worked just as well for them. So that's good. But in terms of the side effect profile, like what we call reactogenicity, it seemed like it was lower in the older people than in young adults. We don't know about teens. There weren't enough teens really. They didn't separate out the side effects among the teenagers. They just lumped them in. But if you look at like how many people have sore arm, fatigue, fever and stuff, it was few percent lower among young adults than it was older adults. So that could mean that as we go down younger and younger, that we see that, in our younger children, the same issue. We won't know until we go down. I know for example, the Janssen study vaccine, for their other vaccines they have in that platform, kids reacted better to lower doses. So like for Ebola vaccine, they could give half essentially the dose that they were giving to the adults, and the kids made just as good or better response. So it may be that we can give a little lower dose and get less reactogenicity, but we don't know that yet.
Great. Well, this has really been great. I know that everybody has to get back to their day, and we appreciate you taking the time out from your time on service. I want to see if there's any last questions. I guess my one last question I had is whether you knew anything about sort of the rollout to one thing that we've come up with is getting a lot of the people, the healthcare providers in our office, vaccinated that don't necessarily have hospital privileges somewhere. And I don't know if you've heard anything from the state, whether that's going to be through local health departments or the pharmacy route. I know that practices hopefully can sign up to be a vaccine site, although that's link isn't sort of up and running yet. We're, I think, maybe next month, but didn't know if you knew anything about that.
Yeah, I don't know specifically where you are, or actually specifically where I live either. But I do know that the first tranche that came was relatively small,
Yeah.
and what's coming this week and next week is going to be very large, meaning there's going to be a lot more access now. I think to start, it will primarily be in public health, either, well, hospitals, of course, long-term care facilities, but then in public, wherever the public health jurisdiction has set up the vaccination sites.
Yeah.
In pharmacies, because they have an arrangement with CVS and Walgreens.
Correct.
And then over time get to practices. Here, there's been not that many vaccinations among healthcare providers yet, but we're expecting very soon, like this week into next week, that we're going to start seeing a lot more. So yeah, they wanted to do essentially like beta testing to start, because the vaccine is kind of tricky in terms of cold chain, and they want to make sure they're not shipping out thousands of doses and then they're getting wasted because somebody couldn't handle them. But then once they could prove they could handle them, then they're ready to ship a lot more to all of those sites. Yeah. So it's soon, and I think I've been telling the state, and I think we in pediatrics, and especially you in pediatrics, should be vaccinated soon. And I know some providers have been able to get vaccinated, and hopefully it will be in the near future. And even though I know we're ending, through Nancy or just through you, Paul, or anybody, if there's specific questions people have, you guys are a part of us. So I can help with anybody within their own personal lives or family or within the clinic or whatever about questions about the vaccine. I'm very happy to do that.
Great. Yeah, that is really appreciated, because as you know, everybody's, circumstance is the same in some ways, but also, a little different. So I just want to thank you for taking the time, and thank everybody for joining, and we'll meet up again soon. Thanks a lot, everybody.
All right. Thanks. Happy holidays and
Bye.
stay safe and talk
Yep.
soon.